Let's first look at a genetic diagram. Let's consider a monohybrid cross between two plants with different height. Both are heterozygous. Tallness is the dominant character, whereas the shortness is the recessive character. There are some steps to follow when you are making a genetic diagram. First, use a capital letter T for the dominant allele and the corresponding small letter for the recessive allele. State the genotype and phenotype of each parent. When writing allele pairs, always write the dominant allele first. After that, label the genotypes parents. Write a small letter X between the genotype of the parents to indicate a cross. After writing down the possible alleles in the male and female gametes, label them as gametes and add lines for each possible combination. Show the results for possible combinations of gametes and label the genotype as the F1 generation and state the phenotype of the offspring below the genotypes. After looking at genetic diagrams, we're going to look at another method called the Punnett Square. This method avoids having many lines crossing each other. There are some steps to follow when you are constructing a Punnett Square. First, you need to draw 4 boxes. Write down the possible alleles in the gametes of the female parents along the top above each box. Do the same along the left side for the male, one next to each box. Label the female symbol and the male symbol on the 2 sides. In the appropriate boxes, write down the possible combinations of gametes when fertilization occurs. Then, state the phenotype of the offspring. There are 3 important points you should pay attention to. First, the outcome of any cross is not related to that of any other. Second, it is possible to predict the proportions of offspring that will have a certain genotype or phenotype. For example, when offspring are selected at random, 3 out of 4 will have 3 earlobes and 1 out of 4 will have attached earlobes. If 200 offsprings are produced from a large number of heterozygous couples, there will be about 150 with 3 earlobes and about 50 with attached earlobes. Third of all, genetic diagrams and Punnett squares only give expected results. If a large number of offspring are produced, the observed results will be very close to the expected results. An organism with a dominant character can be either homozygous or heterozygous. To identify its genotype, we can cross the organism of unknown genotype with a homozygous recessive organism. We call this type of cross a test cross. Let's look at an example. A test cross is carried out to identify the genotype of a tall pea plant. Let big letter T represent the alleles for tallness and small letter t represent the allele for shortness. There are 2 possible cases. The genotype of tall plant is either homozygous dominant or heterozygous. Let's look at the case where it is homozygous dominant. From case 1, you can see that all first phylogenenotype is heterozygous with the phenotype of tallness in all offspring. However, in the case where it is heterozygous, the ratio of the genotype of the first phylogeneration being heterozygous and homozygous recessive is 1:1. From the above results, we can see that all the offspring are tall. The tall plant must be homozygous dominant. However, if the tall and short offspring are in a ratio of 1:1, the tall plant must be heterozygous. Not only did Mendel try to explain monohybrid inheritance, he also went above and beyond in explaining dihybrid inheritance. But wait, what is dihybrid inheritance? It refers to the inheritance of two pairs of contrasting characters at the same time. Mendel crossbred purebred pea plants that had round and yellow seeds with those that had wrinkled and green seeds. By doing so, all plants in the F1 generation had round and yellow seeds. Mendel then followed the F1 plants to self-pollinate. He found out that it results in 4 types of seeds in the F2 generation. They include round and yellow, round and green, wrinkled and yellow, and wrinkled and green. Mendel therefore proposed another hypothesis to explain the results. By carrying out experiments on dihybrid crosses, the prelate came up with another law to explain the result. It is known as Mendel's second law of inheritance, or the law of independent assortment. So what is this about? It states that separating of alleles from one gene is independent of the separation of allele for another gene during gamete formation. Let's try to interpret the results together. A pair of alleles is responsible for controlling the shape of the seeds with another pair of alleles on a different pair of homologous chromosomes. A big letter R represents allele for round seeds and a small letter r represents the allele for wrinkled seeds. Similarly, a big letter Y represents the allele for yellow seeds, and a small letter y represents the allele for green seeds. During gamete formation, to make sure each gamete receives only one allele from each pair, alleles of each gene separate, resulting in each parent producing one type of gamete. And in this case, it's either big letter RY or small letter RY. With the fertilization of gametes, all F1 plants have round and yellow seeds, as alleles for round and yellow are dominant to that for wrinkled and green respectively. In F1 generation, the alleles of two genes are sorted independently during gamete formation, which results in the production of 4 types of gametes: big R big Y, big R small y, small r big Y, and small r small y. With 16 possible combinations of gametes when random fertilization of gametes occur, 4 phenotypes are shown in F2. Now, let us talk about how blood groups are inherited. A person's blood can be classified into group A, B, AB, or O. Blood groups are determined by the type of antigens on red blood cells. There are 2 types of antigens, A and B. There are also 2 types of antibodies in the plasma, namely anti-A and anti-B. Let's look at a table that shows the types of antigens and antibodies in people of different blood groups. People with blood group A will have antigen A on their red blood cells and anti-B in the antibodies in the plasma. People of blood group B will have antigen B on their red blood cells and anti-A antibodies in their plasma. People with blood group AB will have both antigen A and antigen B on their red blood cells. Last but not least, people of blood group type O will have no antigen on their red blood cells, but have both anti-A and anti-B antibodies in their plasma. The production of antigens A and B is controlled by a single gene. The gene has 3 alleles. IA is responsible for the production of antigen A. Similarly, IB is responsible for the antigen B, while small letter I produces no antigen. Genes with more than 2 alternative forms are said to have multiple alleles. Since humans have 2 sets of chromosomes in the body cells, each person can possess only any 2 of these 3 alleles. Alleles IA and IB are equally dominant. Allele combinations IA-IB produces both types of antigens. This condition is called codominant. With allele small letter I being recessive to alleles IA and IB, let's look at a table that summarizes different possible genotypes and the blood groups they result in. With genotype IA or IA and small letter I, the antigen produced will be A and the blood group will also be A. On the other hand, if we have genotype IB or IB small letter I, the antigen produced will be B and the blood group will be B. However, if we have genotype IA-IB, the antigen produced will be A and B, and therefore the blood group will be AB. Last, when the genotype is small letter I, there is no antigen produced, and the blood group will be O. We can use a genetic diagram or Punnett Square to predict a person's possible blood group. For example, a man of blood group O and his wife of blood group AB are going to have a baby. What are the possible blood groups of this baby? As you can see from the diagram here, the baby will have a 50% chance of being blood group A and a 50% chance of being blood group B. This is the end of the video. Thank you for watching. Please give us a like and subscribe to our channel. Click on the tab to watch the next part of the video series.